everyone. I just wanted to spend a few minutes talking to you about the lab report that you have coming up that's due and your project that's coming up. So your lab report is due on Monday, so this is the rubric that I gave you the other day. I wanted to spend a few minutes talking to you about that. So this first part here is the title. Remember, the title is a real easy point to get, but just a couple reminders. Don't just write electrophoresis lab. You need to make sure that you tell me what the whole point of the lab was, what were you testing, what were you trying to find out. Remember, like most of you did a great job on your plant titles, so you were tested the effect of on the growth of geraniums or whatever. So just make sure that this lab reflects that as well. The next part is the introduction. The introduction is using the packet that you got for your pre-lab. So you should not need your pre-lab questions in order to do that because you turned those in and I have those with me. But as long as you have your packet, you should be good. And I told you not to turn that in, so you should still have that. So there's several parts to it. The first one, electrophoresis. So just tell me what it is and how it works. Make sure that you're talking about how you set it up, things of that. It's not just simply a definition, so be pretty descriptive with that. Second part is how DNA moves through the gel. So for that, you're going to talk about two things. You're going to talk about how the charge moves and then the size. So charge and size. Some background information on the Smith family. What do we already know? And then a description of Duchenne muscular dystrophy, which is the disorder that we're looking at for this case study. So when you are writing your background information, please make sure that you're not just writing the answers to the questions one by one. Make sure that it's a flowing paragraph that makes sense, that you've thought it out, and that it makes sense in that paragraph. The next thing that you have to do is the data and the results. This is the data and the results page. So this sheet that I gave you the other day is all that you have to do. So the first part is the raw data, which is this part here. So you've labeled this, you followed these directions, you labeled it, you colored it correctly so that the blue is blue, the purple is purple. And remember, if you had green, you didn't have to draw that because that was just the loading die. So just two colors, blue and purple. If you already have the green on there, that's not a big deal, but just make sure that when you're interpreting your bands, the only bands that are important are the blue and the purple. And then this is the table that you have to complete correctly, this part. So I want to talk to you about this for a little bit because it's worth 16 points, so it's really important. So on the first, telling you what test tube they were in when you loaded it, so you knew who was who. The second row is mother, and it just tells you who it is. You don't have to do anything on this row except for the last one where it says fetus, male or female. You need to circle which one it is. And then the next part is the interpretation. So it tells you that the mom is a carrier, the dad is healthy, and Daniel has Duchenne muscular dystrophy. We already knew that going into our case. You then need to determine, based on the analysis of your gel, so you need to look at your bands and you need to see who it matches up with to determine if they are healthy, if they are a carrier, or if they have Duchenne muscular dystrophy. Those are your three choices, and that's what you will write here. The number of dystrophin genes we talked about in class. So remember that it's a sex-linked disorder, so think about what that means and how many genes the mom has, how many genes the dad has. It's not asking you if they're healthy or if they're normal. It's just asking you how many genes they have total. And then the last part is the genotype. So you should know how to do the genotype. It's a sex-linked disorder. You use their phenotype, or not necessarily, even though with this part we identified if it's a carrier so that you can figure out exactly what the genotype is. So you use this information of what they are that we got from our genetics test to figure out if they're a carrier. Remember that it is sex-linked, so look back in your notes, look back on the practice worksheet so you know how to correctly write that genotype. All right, so that's 22 points right there. So it's really important that you do this correctly and that you turn this in, okay? Then the next part is the conclusion. So the analysis of the data. Basically, you're taking the table and you're telling me what it means. So which family members have Duchenne's, which ones are carriers, which ones are healthy, and how do you know that? So use your data to support it. Two things learned in lab related to real life. So just how can we use this in real life? And then did you have any errors in there? What kind of experimental errors did we have? Did you have mistakes loading? Did you have to get more than one tip per person? And all of that's learning experiences because it's the first time that you did that, but it's still errors that could have influenced your results. So make sure that you keep those, that you list those. The complete sentences actually should have been down here in the formatting. Both the conclusion and the introduction need to be in complete sentences, so make sure that you do that. And then the lab questions. Those are the questions that are on the back of this paper. So just turn it over, answer the questions. They need to be answered in complete sentences as well. If you want it to be really neat, you can retype the questions and answer them on your lab report, or you can write them very neatly here. I will not try to interpret your handwriting because it is a formal lab report. You are allowed to answer them on here, but make sure that it is very neat. If you have very messy handwriting, I suggest typing it. And then just make sure that with everything that it is a formal report, so make sure that you have your headings. You should label everything. You should label the introduction. I had some problems with that on the plant lab. So make sure that everything that is in bold on here should be in bold on yours. So for this one, you only have to have the introduction and the conclusion labeled because that's the only thing that you necessarily have to type. If you type the questions, please put that in bold as well. Okay? So that's worth 57 points. Should be a great grade for you for the end of the year. So make sure that you have good data and that you have that completed for Monday. Okay? Now let's switch gears and talk about your project. So here's your project paper. If you don't have it handy, I'm going to pull it up for you. I think your project is pretty self-explanatory. There's just a couple things I wanted to highlight. You should have a disorder that you wanted to research. If you were absent the day that I assigned this, you should pick up a packet that I left with your name on it. If I forgot to put that with the sub-plans, you can get it. Excuse me. You can get extras out of the bin. Okay, sorry. And then I did leave the list of disorders with the sub. So make sure that you sign up with which project you want. If you're going to join a person that was working by themselves, that's fine. Just put your name next to there so that I know what's going on. Okay? Now, let's look at this project. So we've identified the disorder that you want. And for some reason it's not scrolling, but that's okay. So what I want you to do is that there is a bunch of questions that you need to answer, a whole ton of them. So my advice to you is to either open a Google Doc that you and your partner share with each other if you're doing it with a partner. If you're doing it by yourself, I don't care where you take the notes. But make sure that you have all the answers to all of these questions. There is a lot of them that I want you to know. We're putting together lots of things that we have learned about DNA and genetics throughout this whole quarter, putting it all into one thing. So how was it discovered? Who discovered it, if that's appropriate? How is it inherited? I want you to use a diagram here. That's really important that I want you to use a diagram. So if it's a sex-linked disorder or dominant recessive disorder, you could use a Punnett square. If it is a chromosomal disorder, you can just show me through non-disjunction how that works. And you can probably find diagrams that you can go off of on the internet for that. Physiology of the disease, so how does it relate to DNA and gene expression? Are the proteins wrong? What's going on with the really small biology? Like once you get down to specific DNA, what is wrong? And then some things that are going to affect the person. What are the symptoms? What are the treatments? What are the effects that it has on the individual? Is there additional medical assistance that you didn't mention before that the patient will need, and does it vary based on the age? Does it get worse as the person gets older, or is it pretty much the same? Are there any prenatal tests that people can go through for diagnosis before the baby is born, and if so, describe how those would be done? Identify and describe where in the world the disorder is most prevalent, and if there is a reason for that prevalence, please explain that. What new research is being done? There's tons and tons of research being done, so find out what that is, and are there any ethical concerns that are surrounding the research? There's a couple of the disorders that you may have picked where it's actually a benefit to the person to have the disease or to be a carrier for the disease. So I would like you to look at those and tell me if those exist for your disorder. And what's the hope for the future? There's all kinds of research being done. What do they anticipate a cure coming soon, or that kind of information? And like I told you in class, the only rules I have for your presentation is that it is not a PowerPoint or a poster. You need to be a little bit more creative with this. So you could do a video and link it to YouTube. If you don't know how to link it to YouTube or you don't want to link it to YouTube, as long as you bring in the your video camera and I can hook it up to my computer, that would be acceptable too. Doesn't have to be on YouTube. You can do a web page. For the web page, if you want to use that site that we used for the last time we did a global project where we looked at the human impact on the environment, that was PV Works. You can go there and do that. A museum display, you can do one of those trifold poster boards for that, and then you also have to have some props to go with it. A museum display is never just a poster, so you need to have it be interesting and bring in some stuff that the people may use or may need or to support it. A photo story is the only exception to using a PowerPoint or Google presentation. With that, the only words that can be on your project are the very first slide. And on that slide, all you can have is the name of your disorder and the names of the people that are doing the project. Everything else has to be a picture that is talked about up here. And then you can just explain it as you go through it. And then the last option is a Prezi. Lots of you talked about wanting to do Prezis. So if you haven't ever used it before, it's just Prezi.com. And I'm going to try and pull it up here for you. So you just go to Prezi.com. You have to create an account. And then once you have created an account, you will go here. You can click on, you will create a new Prezi. So I will click on new Prezi, but my computer's working really slow, so we'll see if that works. But anyway, you go on to new Prezi, and then it's kind of like Google presentation. Here it comes. On the left-hand side, you will have a list of all the slides, basically, and then it will show you where you are. You just add in the information. There's all kinds of different templates to choose from on here that you can use it for. So pick one that you think is interesting and that supports your project the best. And unfortunately, it's taking forever to load. So I was going to show you a little bit more of it. But then you can, you're not limited to those choices. That was just what I brainstormed to come up with. That would be a little bit different, a little bit more interesting when we're going to be listening to 15 to 20 of these presentations in a day. Interesting. So that is your project. And unfortunately, I don't know what's going on and why it's taking so slow. But if you have any questions, let me know. I will actually be in class on Monday. I asked to not go on the field trip so that I could be there to help you answer any questions that you have on the project. I will not be there on Tuesday, but I will be there on Monday. You should be getting your review sheet in class today for your test, so work on that as well and let me know if you have questions. We'll spend some of Wednesday reviewing. I would like to start some project presentations on Wednesday, so I'm offering anybody that turns in their project before Wednesday, before 8 o'clock on Wednesday, for 8 o'clock Wednesday morning, if you have your project finished and you have it turned in, I will give you 5 points of extra credit. All right, so hopefully you watched this whole entire presentation and you know that little trick that I just gave you. Many of you have been begging for some extra credit. It's not a lot, but it's better than nothing. So stay before 8 o'clock and you get 5 points extra credit. Have a great day and a fabulous weekend, and I will see you all on Monday.